Hi there! In this video, I will discuss prokaryotic replication. To start, we must first understand the basics. Prokaryotes are single-celled organisms such as E. coli that contain circular DNA and lack a nucleus. Moving on to replication, it is defined as the process by which a double-stranded DNA molecule is able to copy itself and produce two identical daughter DNA molecules. This process forms a part of the central dogma along with translation and transcription. Some important details to remember. In replication, one template strand is used from the old DNA, and one separate strand is produced. Thus, half of the DNA is conserved and half of the DNA is new, meaning that it is semi-conservative. There are also 3 steps to replication: initiation, elongation, and termination, which I will discuss further on. Now, there are several models of semi-conservative replication, but given the circular DNA of prokaryotes, the most common one used is the Theta model. This model contains a singular replication origin, where replication begins. Here, it can proceed in both directions from the origin, causing twisting and making it bidirectional. Thus, 2 replication forks are formed along with a replication bubble. To better understand this process, we'll take a closer look at the replication fork. The first thing to note is that replication can only occur in the 5' to 3' direction, meaning that the bottom strand here is a leading strand created by continuous replication, and the top strand is a lagging strand created by discontinuous replication. This discontinuous process creates smaller pieces of DNA, also known as okazaki fragments. It is also important to label the 5' and 3' ends because it will help determine which strand is which, since replication can only occur in the 5' to 3' direction. There are various other enzymes and molecules that are involved in this process, so we'll take a look at that. The yellow molecule on the far left, known as gyrase, relieves the tension from supercoiling. In green, we see the molecule known as helicase, which attaches at the replication fork and serves to unwind the DNA by breaking hydrogen bonds between the bases. Following is a primase, which lays down the RNA primer since the DNA polymerases require a 3' OH end. The pink molecules known as single-stranded binding proteins then help keep the DNA as single strands, keeping it from renaturing. These next molecules are known as DNA polymerase III, which attach to a primer and add nucleotides. There's also another polymerase known as DNA polymerase I, which removes the RNA primer and replaces it with DNA. Finally, in purple, we have ligase, which is the enzyme responsible for sealing the new strand by repairing the nicks in the DNA with new phosphodiester bonds. This has been the basics of prokaryotic replication in 3. Thank you for watching and stay tuned for more.